to start by introducing our speaker here today. So my name is Adriana San Miguel. I'm an assistant professor in chemical and biomolecular engineering. I'm part of the Genetics and Genomics Academy, in case you don't know me. And today I have invited Eleni Gourgou from University of Michigan to tell us today about her really cool work with C. elegans. I really like her work, and I think we're a little bit similar because we're both engineers working with these little nematodes, which, as you know, are excellent model systems for genetics. So I'll tell you a little bit about her. She did her bachelor's in biology from the National and Kapodistrian University of Athens in Greece. She also got her PhD from this university in animal cell physiology. And then after that, she was a postdoctoral fellow at U Michigan in the mechanical engineering department, as well as the medical school. And she was initially working in microfluidics and neurophysiology, and later on invertebrate animal behavior and the dynamics of biological systems. Right now, she's a research faculty in the mechanical engineering department in that university. And she uses these little worms to study learning, memory, and locomotion. And she does a bunch of really cool approaches that I'm sure she'll tell you about. Something really interesting about Eleni's work is that she's also the associate director for space biology at the University of Michigan Space Institute, where she's leading an interdisciplinary team to build a first three-dimensional interactive prototype of the C. elegans nervous system. She has a KO1 Career Development Award from NIH, and she was also recognized with the 2021 Willie Hobbs Moore Aspire Advance and Achieve Award by the U Michigan Women in Science and Engineering. And so with this, I won't take more of her time. Take it away, Eleni.
All right. Thank you very much, Adriana. This is really a pleasure to be here with you today, and thank you for this opportunity. As I was telling Adriana earlier on, I would definitely prefer to be there in person and get to interact with you all, colleagues, students, everyone there. But I'm teaching a class actually today at 3 p.m., so we had to work around that. And given this constraint, if people are interested in chatting more, and if the Q&A time today is not enough, I have blocked a couple of hours in my schedule for tomorrow afternoon, so I will be more than happy to Zoom call with people if anyone is interested. All right, so as Adriana mentioned, excuse me, the work that I'm going to talk to you about today has been performed in two different sites in the University of Michigan, Mechanical Engineering Department and the Institute of Gerontology at the medical school. And the reason why I'm repeating that is because I think that this is indicative of the interdisciplinarity of the work. And I'm not going to talk about the activities in the Space Institute, but if people are interested, I'm more than happy to discuss more on that topic too. So I just want to make sure that I acknowledge the funding sources for this work, and also people who have contributed or who have helped us over the years. And these are people in addition to the collaborators and students that you're going to see appearing on the slides later on. So to give you the heads up and an idea of what to expect during my talk, first, I'm going to provide some background on the scientific questions and on C. elegans, our model system. And then I'm going to share with you some of our findings, some of our key findings on maze learning in nematodes. Share with you our work on building a mathematical framework that allows us to capture neuronal dynamics, predict nematode behavior, and generate experimentally testable hypotheses. And then I'm going to present to you some of our work on interventions that can protect against aging-driven decline of learning and memory. And finally, I'm going to wrap up by presenting to you some recent advances in the lab with respect to leveraging 3D printing technology to build novel three-dimensional arenas appropriate for work with nematodes. Okay, so let's get to it. The question that drives the work in our lab is how does the nervous system generate behavior? And behavior, of course, can be considered the outcome of the nervous system, while the input of the nervous system is a plethora of sensory cues, both external and internal, meaning stimuli that originate from within the organism's body. Now, behavioral expressions can have a broad range. We are mostly focusing on locomotion, learning, memory, and to some extent decision-making. And today I'm gonna talk in particular about our focus on spatial learning. So what's the neuronal basis of spatial learning? And with that term describes the process by which animals encode information about their environment in order to navigate toward the location of interest at a later point. And more specific questions around which our research revolves are the following. What is the neuronal mechanism of spatial learning? What is the underpinning mechanism? How does this take place? And in particular, how do individual neurons and neuronal circuits contribute to that? And how does sensory input determine and facilitate spatial learning? And is there any aging-driven decline of this cognitive-like function? Intuitively, we would say yes, but can we confirm that? And more importantly, can this decline be prevented or delayed? Based on the fact that we're interested in untangling the underpinning mechanism down to the single neuron level and single gene level, we need to be able to work with a model organism that allows us to do that, that is amenable to interventions and to manipulations that allow us to map specific circuits to specific behaviors. And in addition, since we're interested in spatial learning, we need to be able to work in suitable behavioral arenas that allow for that. Let's keep those things in mind. Now, of course, intuitively, it makes sense that we're interested in aging-driven cognitive decline, but please allow me to provide a little bit more context. So large-scale studies reveal that cognitive functions in humans like long-term memory or working memory decline pretty early in life in middle-aged individuals, even in the fifth decade of their life. And at the same time, there is an increasing fraction of the population, both in the US and worldwide, that repeat significant decrease in their quality of life because of cognitive decline. With the darker color, the darker the color, the higher the percentage of the population that reports such problems. Therefore, it makes sense that there is a pressing need not only to address aging related cognitive decline, but also to delve deeper into the mechanisms and to the phenomena that underpin, into the mechanisms and the phenomena that are responsible for what we observe. So when it comes to the experimental system, as I already mentioned, we need a model organism that would be appropriate for this kind of studies and the behavioral platform. And when it comes to the model organism, we need to work with an animal that actually displays learning, has a rich behavioral palette, that would allow us to make interventions with respect to individual neurons and genetics, that has the ability, that provides the ability to us to draw analogies with humans, and that it is an appropriate model system to study aging. And I will come back to that later. And on the other hand, the behavioral platform needs to be appropriate for usage with this specific model organism, needs to provide spatial cues so that we can study spatial learning. And of course, it has to be compatible with other techniques in the lab and be highly reproducible. Now, when it comes to the model system, there is an organism that has been described as a premier model system for aging research. And this, of course, is the humble nematode Caenorhabditis elegans, or C. elegans for brevity. And it has been the first organism in which one single mutation was found to double its lifespan, which was a finding that trailblazed an entire field of research. And I invite you to remember the name of that mutant, DAF2 mutant strain, because we're going to cross paths with that later on. Of course, C. elegans meets also the other criteria that I listed earlier, the more neurobiology related ones. And a detail that I would like you to please keep in mind is that worms in the lab are normally cultured either in liquid cultures or on the flat surfaces of agar-filled Petri dishes. And this is an agar-based hydrogel that's called nematode growth medium, NGM for brevity. And the only reason why I mentioned this detail is because we're going to revisit this idea later on in my talk. A bonus trait when it comes to C. elegans, in case you haven't heard of that before, is the fact that it is the only animal known to science with an almost fully mapped nervous system. So the entire connectome of the C. elegans is known, and this is something that, combined with the strong genetics tools, make C. elegans a very powerful, let's say, model system and very tractable model system when it comes to both neurobiology and the biology of aging. So a question is, okay, C. elegans have all these properties that are really interesting, but can they learn anything? So we know that C. elegans are capable of learning. This is something that has been studied for, it was discovered in the late 90s, if I remember correctly. And C. elegans are also capable of associative learning. So to give you an example of what they can do, if let's say that we have a worm that has a weak attraction to odorant A, so they are weakly attracted to odorant A. If worms are cultured in food-seeded plates in the presence of A, they make an association between odorant A and the food source. And that results to them developing a strong attraction to odorant A. So they learn to like odorant A much more than they previously did because they made this association with food. And the way that this associative learning has been assessed so far is by using a standard process, a standard type of choice assay, which is performed on the NGM agar-filled Petri dishes. So here's how it goes. Here's how the standard process goes. So on the surface of the Petri dishes, we place a droplet of the odorant A and then a droplet of the carrier control buffer, let's say. And at the third spot, in equal distance from both, we place a number of nematodes. And then we count how many animals end up at the spot A and how many animals end up where the carrier buffer is. And this way we establish whatever attraction is happening between the worms and the odorant A, how much they are attracted to it. Now, that's the baseline behavior, let's say. Now, as a next step, we cultivate worms on food-seeded plates in the presence of the odorant A. And then we repeat the choice assay with the same configuration. And in that case, we observe that many more animals have now ended up where odorant A is, which means that now the preference is much stronger. And the way that people quantify this behavior is by introducing some type of preference index, like a chemotaxis index, because worms chemotax, right? They are attracted to this chemical. And they visualize, what people do is that they usually visualize this behavior either by using a bar diagram or by plotting this preference index. The reason why I'm sharing this with you is because I want you to have an idea of what the standard processes are, or what the standard processes have been in the field so far. And also because I want to tell you that associative learning in the context of chemical cues is evaluated through the process that I described. But in the context of spatial learning, associative learning in the context of spatial cues has not been explored. And we believe that a reason for that is that we don't have appropriate behavioral arenas. The only arena that we've had so far is the flat surface of a Petri dish. So when we decided to embark on this endeavor and study spatial learning in worms, we realized that the standard practice when it comes to other animals are mazes. So from the great variety of mazes out there, we thought that it might be a good idea to start simple because we have to deal with nematodes, right? So we thought we might as well start with T mazes. But this meant that we had to design T mazes that would be appropriate for use with nematodes. So we went for the simplest design possible, and we designed a simple T-maze with dimensions that match the dimensions of a worm. An adult C. elegans is approximately 1 millimeter long. You can see here the functional corridors and some auxiliary areas that provide additional space for placing the worms in the maze or for placing the bait in the maze, let's say. Then we designed and 3D printed a plastic mold with supporting arms that allow it to sit safely on a petri dish. And then we took advantage of this property of NGM because it is agar-based hydrogel, right? The property that's when it's liquid, when it's warm, when it's hot, but it solidifies once it cools down. So we pour liquid NGM in the petri dish where we have placed the mold, and after it solidifies, after it cools down, it solidifies, we pull the mold out, and this way we have ourselves a nice array of T-mazes inside the petri dish, T-mazes that are made of the exact same material on which worms are normally cultured. So it is a material that is known to be nematode friendly. Okay, and by manipulating the design of the mold, we can have ourselves a variety of different sizes and designs of mazes. We can also introduce micro features into the maze, and we can also engineer the surface, and we can also engineer the surface of the mazes so that they can be continuously rough, the default type of maze, or they can be almost featureless or tactile poor. These are the smooth mazes that we will be using in some of our experiments, as you will see later. All right, so how does the worm maze assay looks like? So here is a recording. We place the worm initially at the base of the T-maze, and then we allow it to travel and explore. And then we give it a time window of almost 30 minutes. And then we record the decision of the worm, and whether it has reached the left or the right maze side. Now, as I said, we provide 30 minutes for the worm to solve the maze. If 30 minutes go by and the worm hasn't made a decision, and please note that we record the first decision that they make. So if the worm hasn't made a decision in that time window, we consider this experiment to be inconclusive. And then if worms have escaped the maze, or if they're immobile, or if they're lost, they're not included in the calculation of indices that I'm going to show you later on. Okay. Now, let me share with you some key experimental findings on this maze learning assay. And this is work that started when I was a postdoc in Alan Hsu's lab here in the University of Michigan. And of course, this is work that remains at the core, let's say, of my research program. So the first experiment that we ran was a control experiment, because we had to establish whether worms have some inherent preference when it comes to either of the maze sides, or some inherent handedness, if you'd like. So we tested the population of naive worms that hadn't experienced any maze before, and results show that worms reach either of the two maze arms with almost equal probability, which means that naive C. elegans behavior in an empty maze is completely unbiased. Okay, there is no inherent preference then. Next, we wanted to test whether worms can locate food, i.e. a bait, in a maze. So we placed food bait at one of the maze arms. Now in the schematic, you see food placed on the left side maze arm, but let me inform you that we have repeated these experiments, of course, with food placed on the other side, and the results are similar. So in this case, the vast majority of animals, a percentage that ranges between 75 and 83%, goes for the arm that contains food. Okay, this means that naive C. elegans behavior in a food-containing maze is strongly biased. And of course, this is interesting, but I dare say that it is not surprising, right? I mean, we know that worms can locate food. So the interesting question in this, now, what happens if we take the animals that have located food successfully, in other words, successfully trained animals, and we place them in a second similar but empty maze This is an empty maze. So the question is, will the worms behave the way they did in the control experiments in an unbiased way, or has their training affected their behavior? And the results show that even in this case, even in the case of an empty maze, after successful training, the majority of animals reaches the same side maze arm with the one that used to contain food during training, which means that trained C. elegans behavior in an empty maze is strongly biased, which means that C. elegans experience in a food-baited maze changed their decision-making when they found themselves in a similar but empty maze. But learning is exactly that. Learning is a change in behavior that occurs as a result of experience. So we conclude that C. elegans learn after training, learn to reach a target maze arm in a simple T maze. So this is strong evidence of nematode spatial learning. Okay, hold on a little bit. How is spatial learning achieved in animals in general? Well, there isn't any short answer to this question. But I think it would be fair to say that most animals that have been studied, and these are primarily mammals or maybe even other vertebrates and some invertebrates that have been studied, they mainly rely on visual cues. These are also animals that have a structured brain. So there are specific cells in their brain that process the primarily visual cues, and they form a mental map, a representation of their environment. And then, of course, there are animals that use landmarks in order to find the way to a target location, or they rely on their own body actions, et cetera, et cetera. And there is some variation on how they do it. But this is the basic and common strategy that they share. However, in C. elegans, things work a little differently, right? C. elegans, first of all, they are eyeless, so they cannot rely on visual cues. They don't have a structured brain, and with only 302 neurons, it would be very far-fetched to say that they can form a mental map of the environment in their head somewhere, right? So we believe that our findings invite us to rethink what we know about spatial learning and actually challenge the norm with respect to how spatial learning is understood and defined in the animal kingdom. Okay, but how do they do it? The question remains. And first of all, what sensory cues are they using? Well, first of all, what are the sensory cues that are available to C. elegans? We already discussed about the lack of detailed vision. So from all the other sensory modalities that are available, the ones that are more dominant and the ones that we are currently focusing on are chemosensation, especially detection of volatile compounds, mechanosensation, which manifests itself primarily through touch sense, and proprioception. Now, as a reminder, proprioception is this animal sense that allows animals to sense the location of their body parts. And consequently, it allows animals to sense their body actions. For example, where their limbs are, or whether their body is bent. Okay. How do we assess the participation of those three sensory modalities in the context of maze learning? First of all, through genetics. So we are using loss of function mutations of genes that code for protein components of relevant receptors, which of course, this means that there are mutations that turn off target genes and their functions. And with genetics, we assess chemosensation, mechanosensation, and proprioception. But we also deploy platform design. We engineer surfaces and structural elements of the maze. And this is a manipulation we're using to assess mechanosensation and proprioception. And if I were to summarize some of our findings, I would say, first of all, that using genetics and having the worms and having ourselves worms with impaired chemosensation made us find that this leads to complete lack of food location ability. So they cannot detect the food because of impaired chemosensation. Consequently, they show no maze learning, which means that this is a learning that's triggered by chemical signals and by chemosensation. When it comes to proprioception, when proprioception is impaired, the food location ability remains intact, but there is still impaired maze learning. And let me elaborate on that a little bit. So in order for us to study the contribution of proprioception, we studied two mutant strains. We studied the double strain TRP1, TRP2. This is a strain that has impaired head steering ability. So it lacks proprioception in the head area, let's say. And the second mutant that we tested is the TRP4 mutant strain. This has impaired, this has loss of function of a mechanosensitive channel that is located in a stretch receptor, in a stretch sensitive neuron in the worm's body. So this mutant strain has impaired proprioception in its body. So both these mutant strains, although they have intact food location ability with the magenta index, they have, compared to their wild-type counterparts, they have completely lost their food location ability. Proprioception is the only sensory modality that we have studied that does not affect food location, but does affect maze learning ability. All right. Yeah. And let me expand a little bit more on the mechanosensation as well. Now, for mechanosensation, we used genetics to play with mutants with impaired mechanoreceptors. And I remind you that this is what happens with wild-type animals. And here again, we tested two mutants, MEC4 and MEC10. These are mutant strains with degenerated mechanosensitive neurons. So what happens is that these worms have reduced food location ability and completely lost have completely lost their maze learning ability. The interesting thing is that similar results, we're getting similar results when we're manipulating the platform, and when instead of the default rough surfaced mazes, we use smooth mazes that lack tactile information. In that case as well, we have worms that are traversing smooth mazes and have completely lost their maze learning ability, which means that touch sense is required and tactile input is required in order to contextualize other sensory cues with navigational information, like proprioceptive cues, for example, cues about the worm's body action. And before I move on to describing the overall strategy, let me share with you a piece of information about the role of structural elements in the maze. So we wanted to test whether the structures of the maze, like the vertical elements, the walls, or the sharp edges and corners, provide information that worms can use in order to learn their way in the maze. So here's what we find. Learners, these are worms that learn. I'll be calling them learners. Learners repeat in the second maze the wall or floor preference they showed in the first baited maze. And with that, I mean that if during their trip in the baited maze, if they perform a primarily over the wall or over the floor route, they repeat this wall or floor preference in the testing maze. This is what learners do. But they only do it when the mazes are rough surfaced, because worms that traverse smooth mazes, even the ones that choose correctly, they don't repeat the wall or floor preference. And using genetics, we acquired another piece of information. Mechanosensation is required for this behavior. Functional mechanosensation is required in order for learners to repeat the wall or floor preference, but not proprioception. So functional proprioception is not required for the worms, for the learner worms to repeat the wall or floor preference. These findings combined mean that structural elements of the arena are important in order for the worms to find their way, and are sensed through touch sense, through mechanosensory neurons. All right, so what's their overall strategy and how do they do it? Here's what we think is happening. During training, a worm is placed in the maze and it senses the surface texture and the maze structure through touch sense. During its journey, it detects food, it detects the food bait, and it navigates itself toward that location. Of course, proprioceptive neurons that are activated register information about the worm's body actions. And finally, the worm reaches the food, locates the food, and tastes the food. We believe that's important as well. And in the testing maze, the worm is now placed in an empty maze, but with similar tactile input, texture-wise and structure-wise. And then the nematode repeats key body actions as with the ones that it performed in the training maze by deploying the same proprioceptive circuitry. So the same body actions in the same environment bring the nematode to the target location, to the same side maze arm as before. In other words, the nematode associates bait-derived chemical cues with tactile input, and therefore a behavioral response is evoked, directed locomotion, i.e. chemotaxis. In the second maze, in the absence of chemical cues, the similar tactile input evokes the same behavioral response, now orchestrated by proprioceptive interneurons. And the behavioral response now is again directed navigation, but in this case, it's learning. We're currently working to figure out the details of this mechanism, to identify particular neurons, and to characterize the biochemical pathways that are involved in this mechanism. So this multisensory associative learning behavior that needs a structured environment in order to take place is something that we were able to explore and investigate because exactly we build the appropriate behavioral arena that allowed for us to delve into the particulars. Without the maze platform, we wouldn't have been able to assess spatial learning in the featureless flat surface of a standard Petri dish. And what's the underpinning mechanism? We're interested in how things work, right? So what are the neuronal circuits that steer this behavior? Okay, here is a point where we bring in mathematical neuroscience to join forces with experimental neurobiology. And the reason why we bring in mathematical modeling is because, first of all, we want to confirm that the nervous system of the worm with only 302 neurons, such a task is computationally feasible. And we also interested to unveil how does this happen in a nervous system with so limited resources? I mean, what is the parsimonious functionality of that circuitry? And also, we want to use mathematical modeling to generate new experimental hypotheses, fine-tune our experiments, and make predictions about our system. And before I tell you more about the mathematical model, I want to talk to you about two assumptions that are there. First of all, when the worms are crawling on a surface, they don't crawl on their belly. They lie on one body side. So how can we tell which side is which? The vulva is the opening of the reproductive system, and it lies on the ventral side, aka the belly side of the worm. So if we express a fluorescent marker in the muscle of the vulva, we can tell which one is the ventral side, the belly side, and which one is the dorsal side, in other words, the back side of the worm. Now, the mathematical model assumes that the body orientation of the worm remains always the same during a maze trip, and that the ventral side is always toward the left maze side, and that the dorsal side is always toward the right maze side. And we believe that this is a reasonable thing to assume because in reality, worms can flip, but they don't flip very often. So they only flip when special circumstances apply, for example, when they're very stressed. The second assumption in the model has to do with the fact that the model accounts only for worms crawling on the floor of the maze. So we don't model any worm behavior on the vertical elements on the walls of the maze. We don't model any behavior on the z-axis, if you prefer. So of course, we had to start with defining a maze space, and we had to start with modeling the signal of the food in the modeled maze environment. The food is stronger, the food signal, is stronger as we approach the baited area, and then it weakens as the further we get from the baited area according to a Gaussian gradient. And the next thing we did is to consider a neuronal circuit. That's the model circuit. So let me describe this to you briefly. So the model we consider includes, first of all, a chemosensory neuron that detects the food. Now, of course, we know that in worms, there are several chemosensory neurons, but we have collapsed all the chemosensory neurons for simplicity, and the model includes only one chemosensory neuron that feeds into the interneuron rim, which in turn feeds into a pair of motor neurons, V for ventral and D for dorsal. And these two motor neurons innervate muscles on the ventral and dorsal side of the worm's body. Now, of course, the worm has dozens of motor neurons, but here again, for simplicity, we have collapsed all these dozens of neurons into just this pair of motor neurons. The model includes also these components, IV and ID, implicit components, that are responsible for the oscillating activation of the ventral and dorsal motor neurons. And this is what enables the model to replicate this sinusoidal type of locomotion that you might have noticed in the videos that I showed you earlier. The model includes as well an omega component. This is a neuronal component that's responsible for introducing changes of direction, turns or pirouettes, as we call them in C. elegans. And all these components that I have described to you are there in real animals. All these neurons are there. And the architecture of this circuit is based on other modeling work done by others in the past. What we have added here is an assumed neuronal component, the gamma neuron. Now, I want you to think of gamma neuron either as a single neuron or as a collection of neurons. And gamma neuron is imperative for our model because here's what it does. Gamma neuron receives input from the two motor neurons. This means that gamma neuron keeps track of the worm's locomotion and also of the curvature of the worm's path, as well as of any change in the curvature of the worm's path, which means that it keeps track of any change of direction. In addition, and because of this input that gamma receives from the motor neurons, some of its biophysical properties, for example, its resting potential, might change if there is a change in curvature. Now, gamma neuron also receives some implicit input from the chemosensory neuron, which means that it is kept in contact with the food detecting machinery of the worm. Now, all these things take place when the simulated worm finds itself in a maze with food. But when we put it in a maze without food or in an environment without food, of course, the chemosensory neuron is not there, so none of that is there. And then what happens is that gamma interneuron now sends an output to the two motor neurons. And the output that the gamma sends to these motor neurons is what defines now the curvature of the worm. So whatever change has happened to gamma because of the input of these two motor neurons during training in the presence of food, whatever changes have occurred to gamma, these changes are now responsible for the output of gamma to the motor neurons. And I'm not going to get into any more details. I've already provided more than enough, but I'm going to tell you that the resting potential of gamma changes and it acquires a sign, positive if the food was found on the dorsal side of the worm, negative if the food was found on the dorsal side of the animal. And this is how gamma now determines the curvature through this output. This is how gamma now determines the curvature of the worm's locomotion. This is why we call these connections learning connections. So I won't provide any more details when it comes to that, but I want you to see, I want to show you two simulation videos because I want to show you how the model replicates the training behavior in a training maze in the presence of food, and how it also replicates that the same worm, the same model worm now placed in an empty maze, how it replicates the behavior during testing, how it replicates learning. And this is something that happens. This is something that replicates the behavior, also the sinusoidal type of locomotion, the exploration of the starting point, the interaction with maze elements, and finally ending up at the destination point. Now, the animal here is modeled as a head point, so we consider the animal to be just one single point. It doesn't really have a body here. And what I also want to tell you is that when we run multiple simulations, like thousands of simulations, the percentage of simulated animals that locate food is very similar to the percentage of real animals that locate food. And the percentage of animals that learn, again, is very similar to the percentage of living animals that learn, which means that the dynamics of the model, which means that the model can capture the dynamics of this behavior overall. And what we're currently working on here is we're working on adding a body to the worm. So some body biomechanics will be included. We are working on adding proprioceptive feedback because we want to make it a little bit more realistic. And we're hoping that after doing that, we will be able, and in combination with genetics experiments, we will be able to delve into the details of the dynamics on the nervous system, combining genetics approach and applied neuroscience. Applied mathematics and computational neuroscience. So we're also interested in aging, as I mentioned earlier. So let me tell you a few things about that. Does maze learning decline with aging? And if so, are there any manipulations that we can follow to delay the onset of that decline? Most of the experiments that I'm going to present have been run by a number of very talented master's students and undergraduate students in the lab. And of course, the first experiment that we ran here was done in order to establish whether there is indeed a decline in maze learning ability. So this is what happens. Oh, I miss a star here, but yeah, that's true. We've seen it before. This is what happens in day one young adults. This is a result that I've shown to you in multiple slides already. Now, in contrast to that, middle-aged animals of day 5 and day 7 have lost their maze learning ability. So even though middle-aged animals can still locate food in the maze, they don't maintain the ability to learn in the maze. Now, these are nematodes that are middle-aged, which means that they kind of correspond to humans, I don't know, 40 or 50 years old. I know that's not very optimistic, and I don't want to think about it this way, but the findings are there, and they actually corroborate other findings when it comes to other types of learning that report an early decline of nematodes' learning abilities early in their life when they're still middle-aged. Now, if you remember this long-lived mutant, DAF2, these animals here, the middle-aged, long-lived mutants do show some maze learning ability in contrast to their wild-type counterparts. And this is important because it's not always the case that prolonged lifespan is accompanied by prolonged health span, but this is the case when it comes to this mutant and maze learning ability. So we want to delve deeper into that and see what's the connection with the insulin pathway. And when it comes to manipulations, we tried dietary manipulations. So we tried starvation, food deprivation for 24 hours prior to testing, and this is again the reference case. So if we starve day 1 young adults, starvation doesn't make a difference. It doesn't affect their maze learning behavior or their food location ability. But when we starve for 24 hours middle-aged day 5 animals, their maze learning ability is restored. Now, this is important because we know from the literature that starvation can delay the expression of the aging phenotype. And the question here is, what biochemical pathway is triggered or is upregulated or downregulated, and that affects the learning behavior? So that's also something we're very much interested in exploring further. But I have to admit that the thing that fascinates me more is what I'm going to talk to you about now. So we have found that sensory enriched environments protect against aging-driven decline of maze learning. And here's what I mean. Middle-aged animals that have been cultured for their entire life on this type of plates that are engineered to be three-dimensional with vertical elements, corners, and all kinds of surfaces, these animals, middle-aged ones, they retain their maze learning ability, in contrast to what happens with their counterparts that have been grown on the flat surfaces of NGM plates. And that's really important because we have been working on figuring out what are the sensory modalities here. And if you remember, we found that it is through mechanosensation that they perceive the structural elements of the maze. So we're currently working on figuring out how exactly being exposed to this kind of environment, how it affects the sensory neurons and the nervous system in general and biochemical pathways in general, in order for the worms to be able to retain their maze learning ability for longer. Actually, the fact that worms, the golden standard for culturing worms on 2D surfaced mazes, is something that has been really catching our attention for quite some time, because this is not aligned with what happens in nature. So in nature, C. elegans live in rich three-dimensional environments like rotten fruit and muddy soil, which has nothing to do with the two-dimensional surface of mazes. I'm sorry, two-dimensional surface of plates. So what we've been doing in the lab is that we've been working to harness 3D printing technology to build realistic three-dimensional arenas. And what we've been doing is that we've been converting a commercially available 3D printer that normally extrudes a plastic filament. We've been converting it so that it can extrude directly the hydrogel, the agar-based hydrogel, on which worms are normally cultured. So we can directly 3D print three-dimensional behavioral arenas. And with the first iteration of our machine, the first prototype, we've been able to 3D print really simple parts like, square frames and this type of C structures and all that. And these don't look very complex, but we've used them to run experiments, and we've used them to show that C. elegans actually perceive those parts as physical barriers, and whether they will cross these physical barriers depends on their age and depends on their feeding status, and I'm interested to provide more information if you're needed. And now with the second generation of our machine, the Menalon printer, we've managed to have much better control over the temperature of the hydrogel and over the overall process of extrusion. And we managed to build some more complex three-dimensional arenas. And by leveraging the potential of these arenas and by coming up with a very carefully thought out experimental designs, we aim in the future to be able to unlock the underexploited potential of C. elegans as a model system, deploy the genetics toolbox, and decipher the mechanism of three-dimensional learning and spatial memory. And I'm going to leave you with that. And of course, I'm more than happy to take questions. Thank you for your attention.
Thank you so much, Eleni. That was really an interesting talk. Let's see if we have any questions from the audience. You can raise your hand or type in the chat. I have a lot of questions, so I'll get started in case while people bring in their questions. I guess I was wondering if you have an idea of what gamma is.
Yes, thank you for this question. I wanted to touch upon this, and I was hoping somebody would ask me. So I don't know if I made this clear enough, but gamma, due to its location and due to the fact that it receives input from motor neurons, it monitors the worm's locomotion and the curvature of the path and any change in that. So we believe that actually the model managed to predict the contribution of proprioceptive neurons there, because proprioceptive neurons, if you think about it, this is what they actually do, right? I mean, they are activated because of the worms bending their body and because of stretch applied on the surfaces. So basically what they do is that they keep track of any changes in the bending, any asymmetries, let's say, in the bending, which occur when the worms change the direction of their path. So we believe that this is what the model predicted, the involvement and the requirement of proprioceptive neurons. So the fact that genetics experiments actually showed that functional proprioception is necessary for learning to occur, this kind of confirms, validates the model, if I can say that, and then the two combined point suggest that we should now check the contribution of specific proprioceptive neurons, like DVA, for example, that might be in the worm.
Mm-hmm. Okay. So how do you plan to test that? Like some ablation experiments, or how do you?
Yes, the idea is to use, to silence those neurons. And we're thinking that maybe the most appropriate way for us to proceed with that is not through laser ablation, but maybe through some genetic silencing by either expressing some caspases there and silencing the neuron, or with the histamine chlorine system, if we wanted to do a more like a reversible way of silencing the neuron. Yes, exactly.
Okay. We have a question from Caleb. Caleb, go ahead. You can unmute yourself.
Hi, thank you for the great talk. I did want to ask, I've got two short questions. One, in the 3D arenas, are the worms actually climbing up and around these 3D arenas, or are they just kind of staying on the base of the surface of the plate? How does that work?
You're referring to the ones that were 3D printed?
Correct.
Okay, thank you. So in this case, I would say the short answer would be that they can do any of the above. There are worms that actually climb on the 3D arenas. There are worms that actually bypass them, if I can use that term. And then there is a very small fraction of the population that actually squeeze themselves between the arena and the surface of the agar plate. So they kind of burrow underneath the arena, but this is a very small fraction. So they do explore and they do climb on those ones, but I haven't quantified what fraction climbs on the arenas and what fraction might bypass them.
Right, that's very interesting. Thank you. And then my other one was, is there any evidence of this, the learning passed on to any progeny? Is that something that occurs generationally, or what does that look like?
We haven't looked in that. We haven't explored that. But given the literature, given the work that has been published recently, I believe that that would be very interesting. But we haven't explored it at all.
Very cool. Thank you.
Thank you.
Okay, we have a question from Fred Gold on the chat. He's asking if this is only one trial learning, or is there more learning over trials?
Yeah. Thank you very much for this question as well. So everything that I have presented to you happens after one single training session. Maybe this is why it is short-termed. It is the time frame of retention is really short. About sequential training or multiple training, we are working on this. There are certain challenges that have to do with repeatedly picking up the worms and transferring them into new mazes. so far what we've seen is that in protocols where we are attempting to train multiple times the worms, if we are very careful when it comes to the time intervals between training and so careful so that the worms won't get demotivated after multiple pickups, there is an improvement when it comes to how long they can retain their learning ability. But I cannot provide more information at this point because I don't have conclusive results, because this is work in progress, and I hope this covers the question.
Okay, Mashael, do you want to unmute yourself and say your question?
Yeah, thank you. Thank you. Very, very interesting talk. I'm not very familiar with C. elegans, but I'm wondering if you looked into their microbiomes and if they play any role of them, like in other models where like the gut-brain axis, and then how would that look? And if so, could they be used as models for like looking into the role of microbiome in cognitive behavior?
Yeah, thank you for this. Like the gut-brain interaction, right? I don't know. I guess one really fascinating thing about the maze learning is that now there are so many things that we can do with that. But this is relatively new. So there are so many things we haven't explored. We are focusing for the time being on figuring out the mechanism and how exactly this is happening. But I would be very excited to work with someone who is an expert on the gut microbiome, so that we can explore different microbiomes, different food sources, and all these different conditions that might affect maze learning. And individuality, like why some worms learn but others don't, is also something that could relate to the issue that you brought up, and I would also be very interested in exploring that as well. Thank you.
Okay, we have another question in the chat. Krista Baker is asking, can worms learn the locations of multiple chemicals separately?
Yeah, now, excuse me, if I understand this question, that would maybe mean placing different chemicals in the maze or something like that. We haven't explored that yet. We have tried different attractants. I mean, we have tried a food source like the standard food source, E. coli OP50. We've tried attractants like L-lysine, for example, isopropyl alcohol and stuff like that in the maze. Learning occurs in all these cases. Excuse me, but placing multiple chemicals simultaneously in the maze, we haven't tried it yet. But this is the reason why we have developed this radial maze design. If you remember, it was on one of those slides, and maybe the last one with the video. So I guess we'll use, instead of the T-maze, we'll use the radial maze to explore what you're suggesting. We've thought about this question, but we haven't worked on that yet. Oh, there's one more. Learned avoidance. Have you observed learned avoidance? Negative learning, that is, right? No, we haven't done that. We haven't done that, no. I think there is some sort of negative learning that we have observed when we pick the worms multiple times, transferring them from one maze to the other. From some point and after, it doesn't matter if there is food in the maze, it doesn't matter if they are hungry, they just don't go anywhere. Because I think that they have associated being picked up with the maze environment. So that is not a conclusive, like a concrete result, but it may suggest that what you're asking could be true, and that they could have some negative learning in the maze. So it would be super interesting to pursue that.
Okay, I have one more question, if I can.
Yes.
I was wondering if, because I saw you have the DAF2 knockout, right? And that extends their learning abilities or the age they can learn at. Do you know if there's a correlation between that time point and reproductive decline, right? Because with DAF2 knockouts, you can push reproduction to later days or, you know, to later.
Yes.
So do you know if there's any association there?
Adriana, no, I don't. And I think that this was just one data point, right? I mean, day 5. And of course, day 5, as you implied, it was determined just by how many days, how many 24-hour sets have passed since the first day that they laid eggs, which of course for DAF2 is kind of moved a little bit later. So I think that in order for us to answer your question, we should run a follow-up experiment with worms tested the next day and the next day, and figure out exactly when on the life of the animal and of its reproductive period this decline falls. So I don't know that. I don't know that yet.
I guess the reason I asked is because, you know, we think sometimes that aging kicks in after reproduction's over, right? There is no, so
Right.
I was wondering if there's a, you know, correlation there.
Yeah, right. And when it comes to wild type animals, there isn't any decline in maize learning before day 5. I mean, maybe we have to explore them in smaller windows, like shorter windows. So day 4, for example, they're doing okay, but I'm not sure about late day 4, for example. So there is a decline. So during day 4 and transitioning from day 4 to day 5, there is a decline in the rate of egg laying, like number of eggs per day. And after day 5, the decline is very sharp. But day 5, indeed, they lay less eggs than they do on day 3, for example, when there is the peak. So it is kind of closer to the end of the reproductive period, if that makes sense.
Makes sense. Okay, we're a minute over. So thank you so much, Eleni, for joining us today. It was really a great talk.
Thank you. I hope I didn't bore you. So.
Oh no, it was really engaging. And I think if anybody was interested in meeting with her tomorrow, please just email me and we'll set it up. Okay.
Great. Thank you very much.
Great. Very nice to see you. Bye.
Very nice to see you. Bye.